All right. It's all going to be okay. All right. When it comes to metabolism, yes, I have this on here. It's a key component to your understanding of many upcoming topics. Most students hate metabolism with a passion. It's like, I hated it when I had it before, and every time I've had it before, and I'm going to hate it now too. All right. I get it. I see your point. However, lots and lots and lots of things in microbiology keep coming back to metabolism. So on one hand, this topic is supposed to be entirely review. You should know what happens in photosynthesis. You should know what happens in aerobic and anaerobic respiration. You should know that ATP is the energy currency of the cell. So a lot of this should be review. But I think a lot of you have likely forgotten it. The nice thing about this is that you can find tons of videos on the internet to help you with some of this stuff, okay? In Blackboard, there is a course resources tab under our lecture, okay, our classroom. That class resources has a set of videos that walk you through these different steps. That they were made in-house by different full-time faculty. They walk you through the different steps. You can consider watching those. Most of them are short, 5 minutes or less. Walking you through the different steps. You can go to Khan Academy. You can go to YouTube and search for aerobic respiration. We're going to focus on metabolism in regards to prokaryotes or bacterial cells. The process is all the same, but where it happens inside the cell is different between a eukaryote and a prokaryote, okay? You learned before about the mitochondria and how the mitochondria are that energy powerhouse of the cell. Well, you know bacteria don't have mitochondria, okay? So in this case, we're talking about everything that's happening is happening in the cytoplasm instead. So try and think about where the differences would come up, but you can find lots and lots of different videos, and they're all covering the same exact stuff. Does that make sense? It's all fine.
It's almost fine. If you fail, it's fine.
It's all good.
It'll be fine.
All right. The question I get most often is, why do I care about this? Who cares about bacterial metabolism? Why do I need to know it? Why is it in this class? I'm going into nursing. I don't give a crap about bacterial metabolism, okay? You do, because you are encountering this all the time in your daily life. All the time. See, this is why I drink. Whether you are having some wine, some beer, microbes are involved. You had some toast for breakfast, you got a sandwich in front of you, or some healthier food than that, okay? Bacteria were involved, yeast were involved in making a lot of those components, okay? You know, cheese. How many of you like cheese? Fabulous stuff, okay? The reason you get different flavor profiles in the cheese is all coming back to the different bacteria that are present. Oh my God, it's blowing up. All responsible for the different flavor profiles, okay? Bacteria are responsible for your food in many different ways. You want to have some kombucha? You want to... No, I don't want to do that. Never mind. Kimchi, sauerkraut, good probiotic foods, yogurt. Bacteria are involved in all of these different components, and the products of their metabolism are really important. Let me just tell her I'm in class, and I will get back to you. Stop sending me 5 texts.
It'll all be fine.
Okay. You are getting experience with all their products on a regular basis, okay? You leave some juice in your car and you go back and it's like the gallon is expanding, okay? Or the gallon of milk, you see it and you're like, that gallon of milk doesn't look right, it's blowing up. It's blowing up because products of microbial metabolism are giving off gas, and that's what's causing that to happen. You ever flatten a gallon of milk and come back later? Flatten a gallon of milk, right? Flatten a gallon of milk, when it's empty, and you come back later and you're like, it's no longer flat anymore. Yes, it could be the seal. Chances are it's bacteria that are left in there that are undergoing metabolism and giving off gas as a byproduct, okay? So you are experiencing the products of this on a regular basis. You got nail polish on, you use acetone to take it off. That acetone is actually made by bacteria, okay? You take antibiotics. The reason the antibiotics, the naturally produced antibiotics, the reason those come up is because of this microbial metabolism. They're using their enzymes, they're making that product. So there's lots and lots of different ways in which you encounter this on a regular basis. It's not just about, I'm going to become a nurse, and why do I need to understand microbial metabolism? This is bad. The Bud Light commercial. Did you see, anybody watch Super Bowl?
Yep.
And he was like, listen, we don't have any corn syrup. And they went on and on about the darn corn syrup, okay? It's not that the corn syrup is in your beer. It's not that the corn syrup is in your beer. The corn syrup is used to help drive fermentation to make the beer. So all of those bacteria and the organisms, okay, you got your yeast, you got your hops, the yeast comes in multiple forms, all this stuff. When all of that gets mixed together, they have to go through fermentation to make beer, all right? They need a sugar source, okay? They are, going back to our terms, okay, chemo-organoheterotrophs. So they need organic compounds for an energy source and organic compounds for a carbon source. The corn syrup is providing the carbon and the energy source so that the yeast grow nicely and make beer. So like Miller Lite came out with some kind of, ad that was like, yeah, thanks, but I don't know. They were like, we taste better or something like that, less carbs. I don't know.
Which is false.
The Corn Growers Association got all angry that how could they make this commercial because they're giving corn syrup a bad rap. It's not high fructose corn syrup. It was some fancy advertising. In the long run, it portrayed it was a little bit misleading. The corn syrup is not there to go influencing the taste of your beer. The corn syrup is there to drive production of the beer to start with. See all these things you didn't want to know, okay? You are encountering this all the time, all right? Here's what you need to understand. We've got these organisms, and these organisms need to grow. If they're going to grow, they have to do two major things. They have to make new components, which is known as biosynthesis. If you're going to grow and divide, you need extra components, right? Think about you were a little baby at one point, and now you're a nice grown adult. In order to do that, you needed to make new cells. You need to make new cell components. Bacterial cells do the same thing. It's all part of a process called biosynthesis. You've got to make new compounds. You also have to be able to take the energy and harvest it and then use it. It goes back to our terms from chapter 4, and we covered them in chapter 3 too. Carbon source and energy source. Every organism has got to have a form of those. The carbon source and the energy source are what allow you to make new components and to get the energy involved in making those new components. When you hear this word metabolism, metabolism is just a fancy word that's saying all of the chemical reactions inside the cell are leading to metabolism, okay? You hear someone say, I don't get fat because I have a high metabolism.
I wish.
Right? It's literally just referring to the chemical reactions that are going on inside your body. You've got a lot of chemical reactions going on, lots of metabolism, you're burning lots of energy, okay? Same thing happens if you're like, hey, look, I worked out today, my metabolic rate is higher. Absolutely, because you're trying to replenish all of those components, okay? So the whole point of this process is coming back to carbon source and energy source and what we're doing with them. We're taking the carbon source and we're using it to make new compounds so that cells can grow and divide. We're taking the energy source. We're harvesting the ATP out of it, and then we're using that to make new compounds. Okay? What do we think so far? It'll all be fine.
I don't know if it's gonna be fine anymore.
It'll all be okay. There is tea in here. Nothing else. Mm-hmm. One day we might not be so lucky. For now it's good. All right, so how are we learning this process? The terms are half the battle. So you're going to see all kinds of terms: biosynthesis, metabolism, enzyme, activation energy. You can make some flashcards for the terms, okay? With a word on one side, the definition on another. Just make sure you've got the terms down. That will be half the battle.
How do you make the flashcard?
You won't believe how many people put the term and the definition on the same side to like double up so that you don't use as many cards, and then you're just reading the definition below it and you're not actually comprehending it.
That makes so much sense.
Right? So yeah, it seems silly, like why would you tell us how to make a flashcard and put the definition on the other side? But you will not believe how many we see that are just, you're like, that's not helping you at all.
I think I'm going to start doing it that way now. Because it's going to be fine.
Because it's going to be fine, it'll all be okay.
I might write the term and the thing on both sides.
Right.
I might just
Right,
cut it
yeah.
off. Okay.
Exactly, you could do that.
You're gonna have students when they don't know an answer on the exam, like in the short answer, like instead of writing something, they're just gonna be like, it's all gonna be fine.
I wonder how many exams I'm gonna get that say it's all gonna be fine.
It's gonna
You just
say,
gave
you
me so
already
many
know that.
ideas.
Right. Okay.
What'll happen if blah, 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 blah,
Yeah,
blah,
right.
chemical reaction, it'll all be fine.
I do appreciate a good joke while I'm grading them, like, oh, at least we're laughing about it, it's not crying.
She write the
I'll leave you a couple.
After
No, mine is
See you next
5, it's all
semester.
okay.
Fine.
Yeah.
Fine.
Fine.
Let's get to the end of the semester.
Yeah. Yeah.
Can we get a point per joke?
Right? So we'll see about
Because
that.
I got a lot of them.
You got a lot to come up with? All right, we'll see about that. All right. So, how we are going to get through this, okay? You want to understand what energy is. So this concept of ATP, this adenosine triphosphate, we're making this molecule. We want to understand why cells need it. Again, they all need an energy source because they have to make new compounds and grow and divide. If you have no energy, you're going to die. Think about that, okay? How is it generated? Well, they're taking that energy source and they break it down and then they store it, okay? How do they use it? They make new compounds that grow and divide. When all else fails, your answers are they have to use it as a source to grow and divide, okay? Why are enzymes the key to metabolism? We're going to talk about how enzymes are functioning in pretty much every chemical reaction inside the cell. Without the enzymes, the chemical reaction doesn't occur fast enough to support life, okay? Organisms get bogged down in the minutia of the process. They don't have enough energy to sustain growth, and then they end up dying, okay? You want to understand aerobic and anaerobic respiration as well as fermentation. You want to be able to walk yourself through those backwards and forwards. No joke, okay? Next week, obviously not on Monday, but on Wednesday we're going to do a little task. And I will give you kind of little strips, and we will be ordering this. Okay, can we figure out what comes first, what comes next? Can you take these individual statements and understand how they're going to associate with one another in terms of aerobic respiration? If you can draw it out, you're good to go. You know, start with glucose at the top and work your way through. For photosynthesis, put your energy from the sun on there and see if you can draw it out. If you can explain it to someone else, you've got it, but you need to be able to work through it backwards and forwards. All right. Remember, metabolism is just the summary of all of those chemical reactions inside the cell. Everything is coming back to chemical reactions. So there are two main properties going on here with metabolism, two main groups. You have anabolic reactions, or anabolism, and then you have catabolic reactions, or catabolism. And they're pictured here in yellow and blue-ish, okay? So, metabolism is breaking things down. Anabolism is doing synthesis to put things together. Everything that happens inside the cell is really more of a cyclical process. You take things in, you break them apart, you take their individual components. Components you don't need, you get rid of as waste products. Components you need to use, you put together to make other things. The same processes that are happening in these bacteria are happening in your body. It's all the same stuff, okay? So catabolism is going to be degradative. It's breaking things down. As you break down bonds, you are giving off energy, okay? The energy is then used to make cell structures and components in anabolic reactions, which are used in making cellular components. The reason you eat, okay? We've got some awesome,
Sorry if it smells.
it's good. I see broccoli. Is that cauliflower rice?
No,
Or-
it's potato.
It's potato.
And beef.
And beef. Okay, see, there we go. We've got awesome good protein, veggies going on here. Starch. We've got all these good individual components. When you eat, they all get broken down. Your body breaks every little bit of that apart. And you take all of the energy that's contained within that food and you use it to make all of these extra compounds. If you have extra energy, what does your body do with it? It stores it, converts it into what?
Glucose.
Fat. Okay? So, if you're not using all of your energy, your body's like, listen, I'm not just going to get rid of that. We don't toss that down the drain. We store it. And if you need that fat later, because you're running out of energy, your body breaks it down, and then you use it to make other components. The whole
Then you
concept
have
of
your summer body.
right, then you have your summer body, exactly. Going on a diet, this whole process is all coming back to, if you know, it's all about calories in versus calories out. All you're doing is this, okay? Same thing is happening in bacterial cells. They are eating sugar. They eat the sugar, they break it all down to get the energy out of it, and then they take that and they can use that to make other products inside the cell.
Does
Yeah.
this also happen on a much larger scale in like muscles?
Yeah. Yeah, they're all doing it.
Fermentation.
You got it.
So this is using the body's energy, metabolism and metabolism. But what if, let's say, I'm working on one day and I put, the pre-workout or even B12 in my body, then what happens there? If I keep adding energy to boost up my levels.
You just keep going. You just keep adding, you keep getting that ATP and other components. So ultimately,
Is it like
B12 and the
something-
other things are basically trying to make your body more efficient in harvesting energy. In some ways, you're getting caffeine, which is driving you to stay awake in other ways. If what you're doing is driving ATP production, you're then using that to build all of this stuff. When you end up with too much of that, your body at one point says, okay, we can't use any of this anymore. It excretes what it can't do anything with, and then the extra that it can maintain or convert, it will turn into fat.
So
Yeah,
the-
you've got.
The difference between caffeine and B12, caffeine you're going to crash and burn after a while,
Right.
but B12 you slowly
You
break
slow-
down.
right.
It harvests that energy.
It's harvesting it better.
Oh.
Right. Yeah, too much caffeine is not a. Don't take any of that before the exam. And it'll all be fine.
What about a caffeine pill?
Well.
Caffeine pills are so messed up.
All right.
You would know.
Oh, man, we're on fire here today. Okay. I love this figure because it's coming back to a number of components we've talked about before. So we hit on the four main types of macromolecules back when we were talking about cell structure and microscopy.
There's good.
So everything inside the cell is made up of one of these four compounds. So you've got nucleotides, amino acids, carbohydrates, and fatty acids. So nucleotides get put into nucleic acids, which make, remember, DNA, RNA, they go to make chromosomes, other components. Amino acids make proteins. They get put together to make proteins. Proteins are involved in lots and lots and lots of things inside the cell. They're embedded in the membrane. They function as receptors. They are going to be forming enzymes and helping drive metabolism additionally. Carbs, okay, think sugars, okay, starch, cellulose, other compounds are important in the cell wall. You need these carbohydrates inside the cell. And then finally, the last one are going to be fatty acids, which make lipids, fats, okay? Fats are involved in membranes and storage and all kinds of other components. So everything inside a living cell is going to be one of these four different types of compounds. You need to make all four of these. Producing them, making them from individual compounds is called anabolism. Putting them together is called anabolism. To do anabolism, to go from the building blocks to the macromolecules to the cell structure, requires large amounts of energy. So the reason your cells need energy and the reason bacterial cells need energy is the ultimate goal is to grow and divide and maintain ourselves. We've got to be able to replenish and keep all of these cellular structures going if we're going to do that. The only way we can do that is by completing anabolic reactions, which require energy. The easiest thing for a cell to do to get the energy is to do catabolism, but to start by breaking down sugar. So glucose is the most simple compound inside this cell that they can bring in, break down, make energy out of. Half of what you eat is all coming down to sugar, okay? The sugar is what's going to drive the most ATP production, okay? Bacterial cells are going to feed off of sugar. The main compound in those agar plates and in liquids when we give them to you is all going to be glucose, okay? When they run out of glucose, they die. Or they have to break down something else in order to drive ATP production. So catabolic reactions starting with glucose and breaking this down make the most ATP. Once you make these individual components, they then can be used to make all of those bigger reactions occur. All this is shuffling around individual components, breaking them apart, putting them together to make something else, okay? You ever play with Legos? You get a Lego set, you're meant to make the Death Star, okay? You've seen that one? It's got like 5,000 pieces. It's amazing, like $600, okay? You can make the Death Star, or you could take those 5,000 pieces and you could make all kinds of other things with it, okay? Or if you had a Lego set and you're like, I'm going to take it apart, I'm going to put it back together again, okay? Or you're going to take it apart, you're going to do something else with it, all right? All of everything that you're bringing in, and these cells are bringing in, is all about those little individual building blocks. You could take those building blocks, break them apart, and decide what else you want to make instead. What do I need today? What do I want with it? All right? It's no different than looking in your fridge and saying, okay, this is what I have to work with. What am I making for dinner tonight? You've got the individual components. Now how do you put them together into something that's actually going to be appetizing instead of sitting around with a bowl of cereal every day, okay? Which may be just fine, okay? That's all that's happening here. Catabolism breaks things down. Once you break them down, you get the individual building blocks and you generate the energy. And then you can take those building blocks and the energy and use it to do anabolic reactions to put things together to allow for growth.
When you eat like good fat versus bad fat, like,
Yeah.
does your body decide which fat it wants to use to like use for this stuff, or does it not distinguish between the two?
It depends on how bad the fat is and what you can do with it. Like trans fats, your body is really just not capable of doing anything with those. And
So it just goes straight to like
yeah,
storage.
you just make other, yeah. Yeah.
So I know there are like some like supplements that keep your muscles and like your body anabolic.
Right?
What would happen if like it stays anabolic and like doesn't really have the chance to go back to catabolic?
So you should be doing the catabolic reactions at the same time you're doing the anabolic reactions. If you are trying to drive more anabolism, you're trying to get more efficient with what you're breaking down. You should always have some supply, as long as you're eating, or you've got a fat store that you can break down. Once those run out, regardless of what you're taking, none of that anabolism will occur anymore.
Gotcha.
So they should be relatively balanced and both occurring at the same time.
Gotcha.
Yeah. You're just driving one to be more efficient, if possible. Okay. Uh-oh. There we go. My screen is froze. What are all the components we need to make these chemical reactions occur? So everything that happens inside a cell is in a sequence of chemical reactions. A goes to B, B goes to C, C goes to D. It's never just individual things happening. it's always going to be a string of reactions all happening kind of at the same exact time, okay? So what happens typically is that the starting compound is going to start on one end and it's going to be converted through a series of reactions into intermediates and then finally whatever the end product is of that particular reaction. You will see this term, bless you, precursor metabolites. The term precursor metabolites is simply, it's a product of one reaction that gets used in another reaction, okay? Every single metabolic pathway is going to need ATP, it's going to need enzymes, an energy source, and it's going to need some level of electron carriers, which we'll talk about in a little bit, bless you, okay? So as we start to build the players, in this case, of what's happening with metabolism, we have to keep all of this in mind. We've got to be always referring to ATP and where is ATP going. We have to have enzymes that are involved in that process. We have to have some form of an energy source. If you have no energy source, a cell is going to die eventually. And then finally, you need an electron carrier. Part of the reason we want you to have chemistry before you come into this class is because understanding metabolism and respiration, fermentation, photosynthesis is all going to come back to shuttling around electrons. Don't memorize these, okay? The whole point here is to understand that biochemical pathways inside the cell take on lots of different forms. What you see at the top is just a straight up linear form. A starting compound goes to different intermediates, and then you've got an end product that's produced. You could have 5 intermediates here. You could have 7. You could have 20 intermediates. Whatever begins that pathway is the starting compound. Whatever ends the pathway is the end product. Everything in the middle is called an intermediate. You will also see it described as a precursor metabolite. You could have branching pathways where you get to a point and suddenly they can go down one of 2 or 3 or 4 different pathways. We can use it over here, we can use it here, we can use it over here. This is like you go to Costco and you buy a rotisserie chicken. You know that you get that rotisserie chicken home, you can do like 20 different things with it. It's all about how you want to recombine it, okay? Whatever's left over, I make it food for my dog, okay? I do 5 different things with that rotisserie chicken, okay? It's all about how you use it. Same thing happens in a branching pathway. It says we know we've got one thing in common, but then whatever else happens with it in the end makes different end products. And then another type of metabolic pathway is going to be a cyclical pathway, in which case you're recombining to make all of the intermediates over and over and over again. The starting material enters into this pathway and end product pops out, but it's always regenerating over and over again, okay? Metabolic pathways inside the cell typically fall under one of these 3 components. There's multiple reactions going on at once. Keep in mind, anytime you see a little arrow, that means a chemical reaction is occurring. Every time you see one of these little arrows, that also means an enzyme is involved in the process. Going back to your chemistry. Okay? How are we doing?
Been better, but
It'll all be fun.
All right. Energy for the cell. Energy inside the cell is in the form of ATP. You've heard of this before. But what you gotta understand is that you convert whatever you're bringing in into that ATP, okay? This is looking at photosynthesis. So energy from the sun, plants, for example, do photosynthesis to harvest that energy. It allows them to grow. What do? We eat the plants, and we take the chemical energy, and then we use that to grow, okay? Or divide or to maintain ourselves. So you have to have a constant supply of energy in order to maintain life processes. If you run out of something that's providing energy, your body goes, okay, what can I break down today to harvest the energy from it? Okay? In the case of your body, it will break down fat, it will break down protein, it will then lead to breaking down muscle and other compounds before your body gives up and you die. Okay? Good or bad? But you've got to have some supply of that energy, otherwise nothing happens inside the cell. So organisms are going to vary in how they obtain that energy and in what they bring in to maintain that source. But ultimately it's coming back to this concept of forming ATP. Okay? ATP, in and of itself, is called adenosine triphosphate. It is an adenine molecule attached to a ribose sugar, and then you have 3 phosphate groups that are attached to it. Adenine should kind of look like your nucleotide, talking about our DNA and RNA, okay? Ribose, RNA, okay, remember? DNA has deoxyribose sugar, okay, as its backbone. So some of this looks familiar, and then what you're adding on here are these 3 phosphate groups. Please notice your 3 phosphate groups are negatively charged. These 3 negatively charged phosphate groups are actually held up together by relatively weak bonds. Because you've got similar charges next to one another, they're all kind of repelling to a certain extent. So the energy in this molecule is actually stored in the bonds that are holding on the phosphate groups. So this bond here has got energy in it. This bond here has energy in it. Okay? So when we say energy is in the form of ATP, the energy is actually stored within the bonds holding the phosphate groups together with one another. ATP is adenosine triphosphate. ADP is adenosine diphosphate. If you break off one of these phosphate groups and you're left with 2, it's then called a diphosphate. When you break off that phosphate, you have energy here that then gets funneled into something else. Okay? You can break off another phosphate. Suddenly it's got 1. Now it's known as adenosine monophosphate, or AMP. Okay? This molecule goes back and forth between AMP, ADP, ATP. When we've got ATP, we've built up all of the stored energy that this one molecule can have.
She's calling me from down the hall.
Literally. 3 molecules on here, and the 3 phosphate groups, it's in its most energy form it's going to have. If it needs some energy, needs to break it off, the cell needs to make something else. It loses a phosphate, the energy goes into putting that together. It loses another phosphate, that energy goes into something else. Okay? So energy inside the cell is formed in this molecule of ATP. The way the energy is stored is really in the bonds holding on the phosphate groups. Now, you see this, what's written on the slide? ATP is created by 3 different mechanisms. The mechanism the cell chooses to use is going to be different depending on the cell itself. Okay, so it's created by 3 mechanisms. Phosphorylation means adding on a phosphate group. Think about what we just said. AMP, ADP, ATP. Okay? Adding on a phosphate group is helping to bring energy into this process. So we create ATP by a process called phosphorylation. If we do photophosphorylation, that organism is completing photosynthesis to make ATP. Organisms that do not do photosynthesis are going to use a combination of substrate phosphorylation and oxidative phosphorylation to make ATP. Your cells are doing this as we speak. They are using both substrate phosphorylation and oxidative phosphorylation to make this ATP molecule. Plants, photosynthetic bacteria, are going to do photophosphorylation. All it means is making the ATP. Again, the terms are half the battle here. Here is it, looking at it in a different way. The bonds are what's holding on to the energy. If you break off a phosphate group, you're left with two phosphates here. This energy then goes somewhere, and it's going to anabolic pathways. If you have catabolic pathways going on, they're contributing. You're adding on a phosphate, and they're regenerating this bond. Okay? It's a cyclical process. You go to work. Why do you go to work? What do you ultimately want from your job?
Money.
Money, okay? Experience.
What job do you have?
Good for you. Right? It's coming back to can I get some money? Can I replenish my bank account? You want that Tesla. You already said that. So you
I want a lot of things.
Oh,
but-
got it.
Experience doesn't pay for it.
Experience is not going to cut it. All right, so
No, people that
if you think about ATP as metabolic money, you wouldn't burn money, right? No, you spend it and you go make more. And you spend it and you go make more. It's just this never-ending process of I go to work and I come home and I live my life, and it's all this roundabout thing, okay? That's all that's happening when it comes to ATP, okay? ATP gets broken down to do other things inside the cell. You generate more ATP when you release it in catabolic reactions, okay? I go to Target and, buy whatever it is you buy at Target. Everything. Okay?
Everything you don't need.
Everything you don't need. All right? All that money just goes to pay Target using other things, but I go home with all sorts of lovely stuff. I go to work, and occasionally, 6 times a semester, they add more money to my bank account, okay? And I make more ATP, I get more money, and then I just, can spend it again. I'm not quite that bad, let's be honest. I save more money than it costs, okay? But it's just this roundabout thing. So phosphorylation is adding on a phosphate to help generate that ATP so that the ATP can then be broken down to be used in other spots,
Save
okay?
up for that electron microscope.
Yeah, no, I don't value the experience that much to buy an electron microscope for Columbus State. New car, maybe.
Maybe.
That should be on the list.
Just wait a few
Quickly.
years and the Tesla is coming out for $35 grand.
Okay. I don't know if mine will make it that long. 3 ways of making it. Substrate phosphorylation uses direct chemical energy to make the ATP. Okay? Think of this as you go to work, they pay you, money goes in your bank account. Every time that bank account goes up, you've got more money. Think of that like more energy, more ATP. Direct production of the ATP. Oxidative phosphorylation, on the other hand, in this case we're going to use the energy from a proton motive force to add the phosphate ion to make the ATP. If you have heard of the electron transport chain, that is completing oxidative phosphorylation. It's making ATP, but it's an indirect way of making ATP. I like to think of this as the money sits in my 401k or whatever investments I got. And I take the interest in that I build interest, and that helps to make me money, okay? So it's not directly money for the sake of money. It's building on that and making more later, okay? So oxidative phosphorylation is using what you've got to eventually put it together to make more ATP in the end. And then you've got photophosphorylation, which is taking that energy from the sun and making ATP. So what does phosphorylation mean again? Adding on a phosphate group. Why do we want to add on phosphates? Making our energy. What do we need the energy for? Everything, okay? Everything inside the cell that has to be made is being made through anabolic pathways. And all of those are going to require energy.
Can you have 4 phosphates?
No. Yeah. The way the bonds come in, it gets too unstable, so it should never add a 4th phosphate. It should be limited to the 3. I think it's too unstable and it breaks down quickly. I'm not saying it never exists. I could have to talk to a chemistry friend on that, but it's not stable enough to be maintained like that. So it should never really be around because of how the electron shells work and the orbital is smaller.
So is it ever going to be a couple, once it have only single phosphate?
You can, if you really drive depletion, you can take off this second one and just be left with AMP. Yeah. There are plenty of chemical reactions inside of cells that will use the AMP and then do other things with it. So that is totally possible. This is just looking at one example of one phosphate off, one phosphate back on. All right, check the box. You know what energy is. The next one are enzymes. This one was on that prerequisite exam. This was one of the most missed questions on the exam were the questions that pertain to enzymes, okay? So what is an enzyme? Okay, it's a chemical compound and it facilitates each step in a metabolic pathway. So it assists in a chemical reaction. It's not a starting material. It's not an end product. It's just helping the reaction occur. What happens here is that enzymes are going to facilitate by catalyzing reactions. So catalyzing, just think, speed it up. So they help to speed up reactions, and they do so because they lower what's called the activation energy. And that activation energy is the energy that's required to initiate a chemical reaction, okay? An enzyme helps the reaction occur faster because it lowers that amount of activation energy. It's not a product, it's not a starting material, it's always there in the end. It just gets used to bring it down. Here's the way this looks. Oh, I'll go back to that slide in a second. Energy speaking, okay, in terms of progress of the reaction, you've got the reactants. They have to take on a certain amount of energy before they get made into the products. Without hitting that energy threshold, they never make a product. The enzyme brings down the required amount of the energy in order to make this thing go. So I like to think about enzymes like coupons, okay? Right? So, you get it. You go to Target. Here we go, Target exam, okay? You go to Target, you buy a bunch of stuff. I don't know. Snacks, whatever, toilet paper, whatever it is, okay, clothes. You have to pay them before you walk out of the store. You should, sorry, you should pay them before you walk out of the store. If you're not paying them before you walk out of the store, they're not technically yours. You're not going home with those products without the cops coming after you, in theory, okay? You need to reach that required amount. But if you got a whole bunch of coupons, gift card, whatever you maybe, okay? It brings down the amount that you actually have to pay them. So the more coupons I use, the less money comes out of my bank account, but I still get to go home with my stuff from Target, okay? All it's doing is bringing down that required amount. So it hits it faster, and it gets to occur faster. Without the enzyme, most metabolic reactions inside the cell never occur because they can't occur at a rate fast enough to maintain life. There's just too much energy that's required for those. Remember, the energy, the enzyme that's used in this process is not consumed in any way. If you've ever used a coupon, you scan the coupon, you don't just like tear it up, you don't throw it away. You gotta feed it into some silly little machine, okay? Or you go to Macy's and they take your coupon and they always give you the coupon back and I just reuse it, okay? It doesn't get used. It's not a starting material. It's not a product. It's not broken down. It can be used over and over and over again. So enzymes inside the cell are really cool because they're bringing down the total amount of activation energy, and they can be used thousands and thousands of times. When a cell no longer needs it, it's like, no, I'm done with you, and it breaks down the enzyme and it uses it for something else. But as long as it needs it, it can be maintained and used over and over and over again. There is typically a different enzyme for each and every chemical reaction. So this process of going from starting compound to intermediate A is one chemical reaction. There's one enzyme here. Enzyme A is needed here. There is a different enzyme taking intermediate A to intermediate B. So these typically work in a lock and key fit. It's only on one certain thing, okay? This is, you know, you're trying to buy water and your coupon is for apple juice, okay? It's not gonna work, right? So they're lock and key fits. Each enzyme is specific for that chemical reaction. If you run out of enzyme A, guess what? All the rest of this stuff doesn't happen inside the cell. Not gonna happen fast enough to maintain anything. So all of these enzymes are really important inside the cell to maintain everything.
Would you ever use more than one enzyme to get to the next step?
So typically, each chemical reaction is only going to have one enzyme. You shouldn't have two different enzymes with one chemical reaction. It should end up being two separate chemical reactions. Yeah. Here's another way of looking at the enzyme, okay? The enzyme is made of protein, okay? When all else fails in this class, if I ask you what something is made of, protein is almost always the answer, okay? Enzymes are made of protein. Proteins, remember, are made of amino acids, and they fold, okay? They've got this tertiary, quaternary structure that as the protein is made, it all folds to make all this nice shape stuff, okay? So what happens with this protein is that it's got an active site. You can see the active site at the top. The active site is the physical site where the substrate, or the starting material, is going to bind. When the substrate binds to that enzyme, you then proceed with the chemical reaction. It forms the products get released. This can go back and forth all day long. Enzyme plus the substrate, enzyme substrate complex, enzyme plus the product. The enzyme note is always here. It's never going away, it's never made into something else, it's always found. If the protein isn't quite right, the shape doesn't work, the enzyme doesn't work, okay? So a lot of this is coming back to correct protein shape and correct protein function. Later when we talk about microbial control, we'll talk about how one way that you kill bacteria is to heat things up, okay? When you cook your food, you cook your chicken or whatever it is, you're heating it up. You're actually denaturing all the proteins. You're including all of the enzymes that are functioning to make that cell alive, okay? So enzymes are made of protein. They're controlling all of these chemical reactions because they're serving as a physical site for the starting material to bind to. They're facilitating that reduction in activation energy, which then drives the product to be formed. What do you notice at the end of all these enzyme names? ACE. Okay? Anytime you see ASE on the end of something, it means it's an enzyme, which means it's made of proteins. Okay? So all of these are different types of proteins that are used inside of cells. Okay? Remember, they're all driving individual chemical reactions. So catalase, for example. Catalase actually breaks down hydrogen peroxide into water and oxygen gas. Lots and lots of bacterial cells make catalase. Monday's class. Remember I told you I thought your strep was contaminated? Strep does not make catalase. Doesn't. When you all left and went home, I dumped hydrogen peroxide onto your individual culture tubes. They all bubbled. Which told me the only way they bubbled is because it wasn't strep that was present. There was something else that was there. Okay? And then today in class I got out other plates confirming that it had something else on it. Okay? It's an enzyme. It breaks down hydrogen peroxide into water and oxygen gas. If you've ever dumped hydrogen peroxide on a wound and it bubbles because your cells make catalase. Dump it down the drain and go home, go to your sink, dump some peroxide. It's going to bubble. Why is it going to bubble? Because there's bacteria in the drain that are making catalase, okay? Or use it as mouthwash, okay?
Ugh.
It's not going to taste great, but if you do it and you spit it out, it'll then start to bubble on your teeth, all of those lovely places you got biofilms forming. It's fun stuff, okay? It's all coming back to that one particular enzyme. So each enzyme does something different. Notice here, what you've got is this column here says metallic cofactor. Cofactors can be really important for enzymes because they help maintain the active site of the enzyme. So iron, copper, magnesium, nickel. Think back to chapter 4. We talked about major nutrients that are needed, required elements for our cell, and then we talked about all of these micronutrients that are things. They're needed in small quantities, and we said when you look at your vitamin label, the vitamins are going to have all these ones that are in massive amounts, and then all these ones at the end that are needed in teeny tiny amounts. You need all of those because they function for the enzymes. And without all of those little cofactors, your enzymes don't function properly. So that multivitamin that you're taking is helping you to get all of these different vitamins and minerals. You're using those to make things like enzymes.
It's all gonna be fun.
It's all going to be fine. Okay? What types of enzymes are there? Enzymes can come in different forms. So you have exoenzymes and endoenzymes. Exo, think exit. Exo, exit. Every time. You will understand what's happening here. Okay? So exoenzymes go from the inside of the cell to the outside. They break down components into smaller products that can then come on through. Remember how we talked about the cell wall? And we said the cell wall is kind of like that chain link fence. The only things that can come through are things that can fit through the little holes in the fence. Exoenzymes are needed by bacterial cells because sometimes what you need is sitting outside and it's way too big. It doesn't want to come on in. So instead, the enzymes go out, they break it down into smaller products, and then it can come on in. You ever tried to bring in something inside your house that was too big to go through the door?
Yeah, I end up
Couch,
breaking whatever's right
table, yeah, you either break it on purpose or out of frustration. Or you take the legs off, you do something else, you make it smaller so that it fits through the door, okay? That's all that's happening here in an exoenzyme. It's exiting. Endoenzymes, on the other hand, endo, think inside. Endoenzymes work inside the cell to maintain this process of enzyme to substrate. Lots of these are working for metabolism, okay? So all enzymes come in one of two flavors. They're either meant to work outside the cell or they're meant to work inside the cell. Here's another look at that cofactor. The cofactor is just here to help maintain this active site, this lock and key fit. If the substrate takes on this shape and it has to fit here, but your cofactor is missing, the shape's not right, the substrate doesn't work, and everything doesn't function anymore. So the cofactors are absolutely required. We're going to talk a little bit later, next class, not Monday, but next Wednesday. We're going to come back to electron carriers, and we're going to talk about NAD, NADP, FAD, okay? All of these things that you've learned about before that you hated, okay? We're going to come back to those. They're electron carriers. All right. I got 2 minutes left. Hold on. Look at this. Enzymes are going to function in a very narrow range of environmental factors. Things that affect enzyme activity are temperature, pH, and salt concentration. Think back to chapter 4, prokaryotic growth. Remember how we talked about acidophiles and neutrophiles and temperature, mesophiles and psychrophiles, and you were learning all of those terms, and we said, hey, they need the optimum concentration, otherwise they don't grow. Why? It's all coming back to their enzymes. So if you don't give an organism the right pH, the right salt concentration, the right temperature, their enzymes don't function and they are going to die. Everything we talked about with regard to chapter 4 and these required compounds, required nutrients, required environmental factors is all coming back to enzymes. What happens here for temperature, for example, if the temperature goes over the maximum for the organism, the enzyme denatures. When the proteins all denature, the organism dies. Okay? So raising the temperature denatures enzymes. Remember what we said. You cook something, you raise its temperature, you're killing all the organisms on there because you're raising the temperature, you're denaturing the enzymes. We said if you take your raw chicken and you throw it in the freezer, it's not doing anything to the bacteria that are on there. They're still alive. Why are they alive? Because their enzymes are actually still totally fine. They're not functioning right anymore, but the organism isn't dead. When you thaw the chicken out, it comes back to this state, and now the enzymes work again. Same thing happens with pH. So when we talk about regulating organism growth, and when we talk about all of these different factors that are really important for the organisms to grow and divide, we're all coming back to the enzymes. What are the enzymes made of again? Proteins. Okay? All right. So